My name is Ben Meyer, and today I'm going to be talking about drug pump induction caused by an anti-cancer ruthenium complex called KP1019. Before I begin, I would like to acknowledge the South Carolina INBRE grant for giving me this opportunity, along with Scott Moyrally from University of Iowa for the reporter construct, Doctors Andrea Totaro and Christopher Alvin from Furman University for their programming and bioinformatics advice, and Dr. John Quinn from Furman for statistical advice. I would also like to thank my mentor, Dr. Pamela Hansen. First, a bit of background. KP1019 is a ruthenium-based anti-cancer drug, which is very promising. In early clinical trials, it was able to stabilize disease in 5 out of 6 patients. It is thought to be able to do this through induced oxidative stress and DNA damage. In the Hansen lab, we chose to use Saccharomyces cerevisiae as a model organism due to the number of similarities they share with mammalian cancer cells. In both cell types, KP1019 has been shown to inhibit growth and cause cell death through DNA damage and induced oxidative stress. Furthermore, in both cell types, we can see that ABC transporter expression leads to increased resistance to treatment. In our model organism, we chose to look at the PDR5 ABC transporter, which is induced in response to treatment. Turning our attention to the graph in the bottom right corner, we can see that the absence of this transporter leads to a 50% drop in resistance to KP1019. Our goal of this study was to identify transcription factors responsible for the KP1019 dependent induction of the PDR5 pump. We used a bioinformatics first approach to inform our selection of candidate transcription factors. The analysis was run on a joint transcriptomic and proteomic data set. Looking at this chart, we're able to see the transcriptomic data in the form of RNA abundance on the x-axis and the protein abundance plotted along the y-axis. Each line represents a single gene and is plotted between the abundances before and after treatment with KP1019. Using the gene in the lower left-hand corner of the graph as an example, we can see that treatment with KP1019 caused both the RNA abundance and the protein levels of this gene to increase in a nearly 1-to-1 ratio. Moving over to the orange line circled near the center of this graph, we can see that treatment caused RNA levels to increase, but proteomic levels of the gene actually decreased. And that allows us to see that the gene is not being regulated in the same manner as the first one we looked at. In its current form, the graph is hard to analyze, but if we divide the genes up by their transcription factors and look only at the line's angle, we're able to start analyzing how much of an effect a transcription factor might have, as shown in the plots on the right. Each histogram represents all of the genes regulated by a single transcription factor. The x-axis records the angle of the line, with 0 degrees marking the 1-to-1 upregulation we saw earlier, and 180 degrees representing the 1-to-1 downregulation. By looking at these graphs, we can start to analyze whether the presence of a transcription factor corresponds to specific types of regulation, like we see on the left, where there are clear clusters, or not, like we see on the right, where the data is more spread out. The presence of the transcription factor on the right likely has no bearing on how these genes were regulated, while the left transcription factor, with its tight clusters, likely was involved in regulation. Looking only at the transcription factors of PDR5, we're able to rank each transcription factor by how strongly their target genes clustered, and chose 4 to experimentally test. Those were PDR1 and 3, which are both drug response activators, MET32, which is involved in sulfur amino acid synthesis, and TUP1, a repressor in DNA damage response. Our experimental methods involved the color-based beta-galactosidase assay. This was performed by first transforming a PDR5 LAC-Z reporter construct into yeast deletion strains lacking the target transcription factors. These samples were then cultured and treated with KP1019. Following that, baseline absorbance readings were taken from the samples. Each sample was then treated with OMPG, which reacts with the reporter to turn yellow. Absorbance readings were then taken at that wavelength and used to calculate beta-gal units for each sample. These represented the abundance of the PDR5 transporter discussed earlier. These values were then plotted and analyzed. The graph on the left, we can see that this experiment was run with both a wild-type strain and a DUN1 deletion strain. DUN1 was chosen as a regulator of TUP1, which was identified with the bioinformatics work earlier. As with other DNA damaging agents, we hypothesized that KP1019 would activate DUN1, resulting in the increased expression of potential TUP1 targets, including PDR5. We would predict loss of DUN1 would block PDR5 induction. Looking back at the figure, we can see two colored columns. The lighter blue column represents a group that was not treated with KP1019, and the darker column shows the treatment group. Along the y-axis, we can see the beta-gal units that we calculated on the previous slide. Starting with the wild-type strain, we can see how the PDR5 transporter is induced in response to KP1019. The baseline levels of PDR5 appear to be higher in the deletion strain, but were not statistically significant. Next, we tested MET32. This regulator is involved in sulfur amino acid metabolism, including that of cysteine. Cysteine is an important component of glutathione, a cellular antioxidant, which could work in opposition to KP1019's oxidative effect. Looking at the data, however, we can see how both strains are nearly identical and show similar induction in response to KP1019, indicating that MET32 is likely not involved in the regulation of PDR5 in response to this drug. Finally, we turn our attention to the PDR network. Because PDR1 and 3 are paralogs, we ran this experiment not only with PDR1 and 3 deletion strains, but also with a double deletion strain. Using a student's t-test, both the PDR3 and double deletion strain showed significantly lower baseline expression of PDR5, and the double deletion strain showed significantly lowered induction of PDR5 after treatment. A two-factor ANOVA also showed significant interaction between genotype and treatment in the double deletion strain. This leads to the conclusion that the PDR network is heavily implicated in the KP1019-dependent induction of PDR5. This brings us to our key findings. Firstly, we found that computational analyses can be used to identify candidate transcription factors and help guide the design of experiments such as this. Secondly, we found that the PDR network is likely regulating the induction of PDR5 in response to KP1019. Loss of PDR3 reduced baseline expression of PDR5, and the dual deletion strain did not induce PDR5 in response to treatment. Future studies could look at the mechanism by which PDR1 and 3 are regulated by the drug, and how this mechanism in yeast could be related to analogous mammalian cancer cell components. I would like to thank you for taking the time to listen to my presentation. And if you have any questions, please feel free to reach out to me by email at ben.meyer@furman.edu.